This is the third of a couple of, well, third of 3 training sessions we've been running over the past couple of weeks, and it's an introduction to Flapjack. So Flapjack is a tool for graphical genotyping, and Ian and Malcolm in particular are going to be talking over this today. This is part of the Templeton World Charity Foundation project that we've got running just now with grass, pea, and finger millet. But we'd also like to welcome anybody who's come along from other projects. So I know there's some bold partners and things here as well. So without further ado, I'm going to pass over to Malcolm Macaulay. So Malcolm is a barley scientist here. He's responsible for a lot of the genotyping work that goes on here. So he's ideally placed to talk about some of the stuff that you can do with Flapjack. So Malcolm.
Thanks. I'm going to try and share 2 screens here, so hopefully everything's going to go okay. Which screen can you see, just to check?
Your PowerPoint.
Perfect. Excellent. So, yeah, welcome to this training session. We'll be looking at Flapjack. As Paul said, it's a multi-platform application which provides visualization and analysis of high-throughput genotype data. So, training summary. Today, I'll go over the background and development of Flapjack, how to install and download the application. We'll look at how you format your input files, how we import our data into Flapjack, some data navigation options. We'll look at some color scheme and highlighting visualization options. We'll look at how we can sort our data, how we can select, excuse me, and filter lines and markers. And finally, we'll look at some initial analysis steps, maybe create a similarity matrix, create a dendrogram, and hopefully a principal coordinate analysis plot. So firstly, we'll maybe look at the drivers behind the development of Flapjack. So in the mid to late noughties, there was a huge increase in genetic information available to researchers. And this was driven by the introduction of next generation sequencing technologies, which drove down the cost of large sequencing projects. So this graph shows the costs of sequencing a human genome and how it's reduced from over $100 million 20 years ago to well below $1,000 now, and we're probably heading towards a $100 genome in the next few years. And we can see where next generation sequencing technology was introduced around about 2007 and its associated effect. But of course, whole genome sequencing is not always required, depending on the questions you want to answer for your project. So genotyping platforms, both low and high throughput, were developed, and they were developed to help reduce genomic and data complexity for researchers. And this led to the requirement of to develop software to handle large data sets that were now available to researchers. So dealing with genotypic data before Flapjack was very awkward and slow. It would normally involve trying to visualize data in Excel, coloring cells and sorting lines and markers with very basic options. So there was really a requirement for high throughput graphical genotyping software which could visualize large numbers of markers and individuals. So this led to a team from the Information and Computer Sciences Department at the James Hutton Institute here in Scotland to develop Flapjack software, and the associated publication came out in 2010. So Flapjack, as I said before, is a multi-platform application providing interactive visualization of high-throughput genotype data, allowing for rapid navigation and comparison between lines, markers, and chromosomes. Since its release, Flapjack has now 11,000 users worldwide, and the publication has over 134 citations. And I'll end our Flapjack introduction section with a wee fun fact. In the UK, a flapjack refers to a sweet tray-baked oat bar most commonly made from rolled oats, butter, brown sugar and golden syrup. And it's a shame this isn't in-person training, as I would have baked flapjacks for everyone and you could have tasted how delicious they are, but maybe we can do that sometime in the future. So introduction over, we can now time to focus on Flapjack downloading and installation. Flapjack is available to download from the James Hutton Institute Information and Computer Sciences download page, and 3 different versions are available. So Windows 64-bit, Linux 64-bit and Mac OS. And during this training session, we'll use the Windows version. So the Flapjack download process is fairly straightforward. Select your preferred download location, then follow the instructions provided. And Flapjack does not require administrator privileges to install or run. And once installed, the home page is visible and we're now ready to get our data in the correct format for inputting into Flapjack. So let's go through the input files required. They should be created in tab delimited format, and the examples we'll look at have been created in Microsoft Excel. So there are 4 different file types. The first file type is called the genotype file, and the genotype file should contain a column with your sample name, a row containing your marker names, and these marker names should match the marker names in your map file, which we'll come on to next. And very importantly, allele scores from your successful assay. It should also contain a header line hashtag FJ file equals genotype. A map file should contain a column with your marker name, a column with a chromosome designation, and a column with a physical or genetic marker location. And you don't have to have your marker or your location names in order. Flapjack will order this for you. And also a header line hashtag FJ file equals map. So a map and genotype file is all that's required to create a Flapjack project, but additional information such as phenotypic and QTL data can also be added to enhance your analysis. So a phenotype file, they should contain a row of sample names, and these should match the sample names in your genotype file, and columns which contain your phenotypic data. You know, they could be yield or disease resistance, whatever phenotype you're working on. And the phenotype file should contain a header line hashtag FJ file equals phenotype. The last file we'll look at is a QTL file, and that has to contain a minimum of 7 columns. Column A will contain your QTL name. Columns B and C refer to the QTL chromosome and marker location, and this must match the marker information in your map file. Columns D and E, that's the minimum and maximum position and the range where your QTL lies in your map. Column F is the trait name. Column G refers to the experiment where the QTL came from, and additional information such as the probability effect of your QTL can also be added. And it should also contain the header line hashtag FJ file equals QTL. So once we have our files created in Excel, we use the Save As option in the File menu, and select Browse to choose the file location, and then select Text, tab delimited from the Save As Type menu, pull down menu, and now we're at the stage where we can create a Flapjack project. So we're at the stage of importing our data into Flapjack, and to say at this point I'm going to try to flip between PowerPoint and Flapjack, so wish me luck on this one. Can you see Flapjack now? Excellent. So from Flapjack's home screen, there are 3 options for importing your data. Under the File menu, there's an Import Data option. Under the ribbon icon tab, there's also an Import Data option, and also under the Getting Started area in the home page, the Import Data option. They all take you to the same window. So using the Browse options, we can import our map file, and again import our genotype file, and then click Import Maps and Genotypes. And once we have the two basic map and genotype files, these are the only two files that are required to create a Flapjack project. But as I mentioned, you might want to import extra features. So go back to the Import Data option and check the tabs along this window. So we can click on Phenotypes, click on Browse, import your phenotype file that we've created. Flapjack will display a summary window. For example, it'll tell you the number of traits you have within your phenotype file, the number of data points. And the last import option is our QTL file. Again, Flapjack will display a summary of the information within your QTL file. So now we have all our data into Flapjack. At this section, we can familiarize ourselves with our output and learn how to navigate around our data. So we'll look at the genotype view, the chromosome view, the navigation mode, marker mode, and line mode. So Flapjack's default view, if we look in the datasets window on the left, displays an allelic grid with each nucleotide attributed a default color. Lines in germplasm run horizontally across the screen. Markers and loci run vertically. During this training session, we're going to look at some barley data. If you look in the datasets window on the left, Flapjack displays a total number of genotypes and markers in each project on the left-hand side. So Flapjack's telling us there are 222 genotypes with 1,605 markers. So how do we navigate our way around this data? Alleles are displayed per chromosome. Obviously, the chromosome information depends on the information you put in your map file, but there's a chromosome pull-down menu at the top. Barley has 7 chromosomes, and we see the option to display each chromosome. We can look at chromosome 1 here. And it's possible now to zoom in and out of your data with a zoom function at the bottom right-hand side of the main window. So we can zoom right out so we can see the full chromosomal information from chromosome 1 for the whole data set, or we can zoom right in to see very detailed data. Once you've zoomed in, you can use the scroll bars along the bottom to look across the line information, and then the scroll bar on the right-hand side to look up and down the loci information. It's also possible to move around the graphical interface using the overview function on the bottom left-hand side. There's a red rectangle which you can move with your cursor, which will move around the genotype view data. When you move your cursor over the data window, Flapjack will display information. If you look at the bottom of the window, it'll display line, marker, and genotype for one specific area that you have your cursor over. So for example, I'll stop my cursor there, and at the bottom, Flapjack tells me that the line currently selected is Sonia, a barley variety. The marker is 11_20798, and the genotype at that specific point is a T. So now we can start looking at a few visualization options. Under the View menu at the top, there are 2 options. Genotypes view is default, and that's what we've been using. There's also the option to look at the chromosomes view. Chromosomes view, that will display a graphical representation of each chromosome you have in your map file with a name, a number of markers, and the chromosome length displayed. And if you click on an individual chromosome, it reveals a marker density heat map below, which will show you where you have obviously a larger number or smaller number of markers along the chromosome. So we'll go back into Genotypes view. And under the Edit menu, there are 3 operating modes available. Navigation mode, which, as the name suggests, allows us to navigate around our data, and that's what we've been doing. Marker mode allows us to work with our markers, and we can deselect or select markers in this mode. And under the Edit menu, we can change to line mode, and that allows us to deselect or select lines. The first time you use Flapjack, a warning message will appear alerting to the fact you're moving between different modes, and this can be easily disabled with a clickbox option. Phenotype and QTL data can be viewed in the trait option, and this is in the datasets window on the left-hand side. So if we click on trait data, and we've got two tabs which we can move between. So we'll look at our phenotypic traits, and the option of colouring our cells using a heat map value can be selected or deselected at the bottom. This can be quite a useful feature if you're dealing with, if you want to check the effect very quickly of your phenotype. If we click on the QTL tab, it shows us all the information that we've imported from our file. So in default view, Flapjack shows us, go back to default view, Flapjack shows us a heat map display from the first three phenotype traits on the left-hand side of the main window. So we can see, where I have the cursor just now, we can see there's spring-winter habit, for example, the number of rows in terms of 2-row and 6-row, the fertility of the ear and barley, and also the year of introduction. So Flapjack will display the first three phenotypic traits on the left-hand side of the genotype window. QTLs are displayed above the marker above the loci information, and if you click on a QTL, it will reveal the name, and it will also highlight the markers that are involved in the QTL region. So we've very quickly covered navigation and familiarized ourselves with the main Flapjack features. So now we'll look at a few visualization options. Look to overlay genotypes, look at the few color schemes that are available to us, and also how we can highlight different types of data. So we'll start this section looking at the visualization menu and the overlay genotypes option. Default, the genotype is overlaid on each of the different allelic tiles, but that can be switched off. And you can see the allele scores have disappeared from our tiles. It's purely a user-defined option. Under the color scheme option and the visualization menu, there are several different options available. I'm not going to go through them all just now, but you can do it at your own leisure. But I'll pick out a couple of particularly useful options. The first one is similarity to line genotype match. So if we select similarity to line genotype match, and then Flapjack will ask us which line we want to compare our data to. So I'm going to use a barley variety called Proctor, and click OK, and I'm going to zoom out to see the whole chromosome. So in this view, all alleles, or all tiles which are green, match the Proctor genotype. All alleles which are red do not match the Proctor genotype. So we can see very quickly the regions of the genome which are similar to Proctor's and which aren't. So this would be useful in a mapping population, for example. Another option, if you're interested in looking at rare alleles or your allele frequency, under the color scheme option, select allele frequency. There's a slider which you can move from the right to left, and obviously the further you go to the left, the more stringent or low the allele frequency threshold is. And as you move the slider to the right, we can start seeing the lower frequency alleles appearing, and then all the way to the right-hand side, we've got all our alleles present. So it's a very quick way to check your rare alleles and low allele frequencies. So go back into the normal color scheme, which is nucleotide model. So now we can have a quick look at sorting our data. We can sort our data alphabetically, by similarity, also by trait, by import order, and we can also manually sort our data if that's something that is of interest. But don't worry, if you sort your data and you get lost, the undo option is always available. So I'll zoom back in to our data so we can see the the line names on the left-hand side. So under the analysis menu at the top, we have the option to sort lines. Say the first one's alphabetically, and we can see the lines have been sorted A to Z. And then the next option is by similarity. So we click this option. Again, Flapjack will ask us which line we want to compare all our other lines to. We're going to use Proctor again. And it gives you the option of selecting which chromosomes you'd like to use for this sorting. So I'm going to select all the chromosomes, all 7 chromosomes, and click OK. And hopefully you'll be able to see Flapjack gives each line a score, with 1 being the most similar and 0 being the least similar. And we can see Proctor is 100% similar to itself. There's the most similar line in this data set is called Mala Abed, which is 86% similar, all the way down to, I think the least similar is 41% similar, 2 lines called Perican and Amarena. So this is a useful feature if you wanted to check, for example, if you've got duplicates in your data set. So we can undo that. So the next sort option is by trait, and this obviously goes from your QTL file, and we can sort by habit. So it's spring and winter type in this case for barley, and it's sorted the data with all the spring lines at the top and all the winter lines at the bottom. And it's also possible to manually move your lines around this data set. If you hold the control key down, and then click on somewhere in the genotype window, you can see I don't know if you can see I've got Triumph, and I'm moving it, a line called Triumph, and moving it up and down the display, and you can manually move your lines around. Another option is if you want to go back to the original order, you can sort your lines by your importing order as well. Flapjack will display a window. It'll ask you to find the file used for the original import information. That's your genotypes file. And if you open import that, it will sort everything back to the original order. And as I say, the undo option is always available. So that's sorting our data. So the next couple of sections, we're going to look at how to select our lines, how to show and hide lines within our project, and how to filter different types of lines, heterozygous, homozygous, and missing lines. So for example, it might be necessary to remove poorly performing samples. Maybe your samples have got low DNA quality within your assay, or and Flapjack has a few different shortcuts to help us do this. So what's important to remember during this section, that as default, Flapjack automatically selects all lines and markers. So under the Edit menu, you can use Select Lines option to select and deselect all lines. So as I mentioned, everything's selected just now, so we can select no lines and go back in and select all. If you just wanted to make a quick selection of a number of lines, you could select none and then select the number of lines you wanted to look at. If you wanted to remove lines from the view but not the project, then the Show Hide Lines option is really useful. You get 2 options. It'll ask you to hide the lines that are not currently selected, and it'll give you the number of lines that involves, or hide the lines that are currently selected. So if we just wanted to look at the lines that I have highlighted there, I would use the Hide Lines That Are Not Currently Selected option. Click OK, and that just leaves us with the lines that I selected. And go back into the same option to restore hidden lines as well, to bring everything back into the project. So the next option involves filtering lines. So I'll select all the lines again. So under the Filter Lines option, we've got 3 different options to filter. We've got missing lines, heterozygous, and homozygous. And this is what I was talking about before. If you had poorly performing lines in your data, if you select filter by lines, then you get a percentage slider. The further the slider is to the left, the more stringent your filtering is going to be. So for example, if your downstream analysis program would only handle data with, for example, 10% missing data, you could click the slider on 10% and ask Flapjack to use the filtering option. It will tell you how many lines have been filtered from your project. And again, heterozygous and homozygous lines can also be filtered. For example, barley, especially the lines we're looking at here, are all varieties, so we wouldn't expect much heterozygous data, probably below 3%. So we could set the threshold to 4% heterozygous data, and it would remove anything that wasn't a pure variety. And Flapjack will tell us how many lines it's removed. So next we can go through the same process with our markers. We can select and deselect markers, show and hide markers, and filter by heterozygous, homozygous, and monomorphic markers. So we need to select marker mode. And under the edit menu, we'll use the same set of rules as we used for the lines. You can deselect all the markers and only select a few, or for example, select your markers in your QTL. If you just wanted to look at a few, and you can remove markers by the show hide marker option. Again, Flapjack will ask you if you want to hide the markers that are selected or are not selected, depending on what you want to look at in your study. Just remove the markers, for example, under that QTL. And there are other filtering options available. You can filter missing markers either for the whole data set or by line. Again, missing markers, this could be in terms of markers which haven't performed well within your assay. Certainly something I've seen within Infinium assays and also Affymetrix data, where you have some SNPs that perform better than others. And again, if you move the percentage slider further to the left, the more stringent your filtering is going to be. So for example, you could remove all data which has 20% missing data and ask Flapjack to filter that out from your data sets. But a particularly useful filtering option is to filter out your monomorphic markers. So if you were carrying out mapping or association studies where only polymorphic markers were useful, asking Flapjack to remove all the monomorphic markers from your data set can really help in this study. So when you ask Flapjack to filter monomorphic markers, you get the option to use specific chromosomes. I'm going to select all and filter, and then Flapjack will tell you how many monomorphic markers have been filtered from your project. In this case, there was none, so this is pretty polymorphic data. So at this point, we've filtered out all the poorly performing lines and all the poorly performing markers and also all the monomorphic markers. But at any stage, you can export the data, the filtered data. Under the File menu, there's an option for Quick Export. You can browse to your location you want to save your files, and Flapjack will export a map file, a genotype file, and the associated phenotypic and QTL file for all your filtered data. So now we're going to look at some analysis options. We're going to look at potentially looking at creating a similarity matrix, a dendrogram, and principal coordinate analysis. But Flapjack also offers the options, different analysis options. It can offer the marker-assisted backcrossing, which allows the calculation of a recurrent parent and the percentages involved for each of the lines across a chromosome. And Flapjack can also display linkage drag and QTL status information. Flapjack also has the option of a forward breeding analysis option, and this translates trait marker genotyping data into the presence or absence of QTLs. But this requires an additional input parameter which can be provided via a GOBI QTL formatted file. There's also the option of indexed forward breeding, and Flapjack calculates the breeding value and weighted breeding value along with named QTL and genes from provided genotypic data. There's also a useful F1 function if you know the parents, and it provides key statistics to determine whether a putative F1 sample is a true F1 cross. And you can also verify pedigrees using the parental contribution, and Flapjack provides, again, key statistics to determine whether a sampled line is a progeny of the expected or designated parents. Hopefully we'll look at some of these analysis options in future training, but as I mentioned today, we're going to look at similarity, creating a similarity matrix from our data, hopefully create a dendrogram and also a principal coordinate analysis plot. So within Flapjack, a similarity matrix measures the genetic relatedness of our samples, and Flapjack does this by comparing the alleles of each line and awarding a score of 1 if there's a 100% match, 0 if there's no match, sorry, and 0.5 if there's a heterozygote present, and 1 allele matches an allele present in another sample. Missing data is not used, and scores for each line are totaled and divided by the number of markers that are used, and a similarity score is created. So under the analysis menu, we can go to create similarity matrix. Flapjack will ask us which chromosomes we want to use for the analysis. We'll say select all and ask Flapjack to create a similarity matrix. So there are 2 visualization options available in the similarity matrix window. A heat map shows the patterns of similarity where the darker blue colors represent a lower level of relatedness. And there's also an option to look at the tabular data. And this reveals similarity score for each of the line. This table can be exported into Excel or other downstream packages for further study. So the next stage of our analysis is to create a dendrogram. So go back to the analysis menu and ask Flapjack to create dendrogram. So the following warning will appear, and it will warn us that this is an experimental feature, and it relies on having an internet connection, as dendrograms are rendered and carried out in the James Hutton Institute servers. So let's hope they're firing on all cylinders today, and I'm sure it'll render quite quickly. Flapjack uses a modified version of the Jacquard index similarity scoring for dendrogram creation, and we can see from the resulting tree from this data, there's a major split between, and this split comes from a growth habit, spring and winter type, in barley where winter lines require vernalization to initiate flowering. So once you've created your dendrogram, it can be exported as either an image or a PDF file. Principal coordinate analysis, we click back on our matrix and go back into the analysis menu. We can ask Flapjack to create a principal coordinate analysis plot. This is done using other ICS software called Curly Wurly, and we get the option of selecting the number of coordinates we want in our plot. Again, this requires internet access, so hopefully it won't take too long, but once the window appears, we'll be able to see. I have to change the window actually again. Hopefully you'll be able to see the principal coordinate plot. And again, this is split into 2 different areas where you've got spring barley on the left and winter barley on the right. And Curly Wurly allows you to rotate the axis. So if you asked Curly Whirly to...
We're not seeing that, Malcolm, sorry.
Are you not? Oh, no.
The wrong screen.
Is that not it? No.
We're still just seeing Flapjack.
No. Oh, okay. Hold on. It's worth seeing. Just bear with me.
That's it.
Excellent, thanks. Yeah, so we have our data split into two, where we've got spring barley on the left and winter barley on the right. And Curly Whirly allows you to rotate the axis if maybe you've asked for more different points on your plot. But it's a cool feature to be able to visualize your data. So we've had a very quick overview of a few Flapjack features, and I'm sure everyone's at different stages in their Flapjack journey, but I just wanted to point out a great resource. And if you require assistance using Flapjack, then the help menu is super useful. It's divided into different sections, and you can also search by specific keywords. And as well, there are training videos available on YouTube, which I think Paul is going to mention afterwards. So finally, I just want to thank the Flapjack development team and acknowledge all our generous funding bodies, and especially like to thank you for your attention today. Thanks very much.
Thanks, Malcolm. That was great. And thanks for doing that. Does anybody have any questions? Aladdin, you're first.
Yeah, thank you for the nice demonstration. So I would like to ask the size of the data, genotype data that maximum we can include. You know, now with the genotyping, we receive a huge amount of markers. So is the capacity of the software is limited to the Excel, for example, file, or we can use we can introduce it from the text file to the program. So the second is that is there any permutation for the missing data to be recovered? Is this also possible in this software? And also, what about the heterozygotes, homo and heterozygotes? When maybe I couldn't recognize how the form, because most of the data you showed that it was like G or A, or so how this could be handled. And the last question on the dendrogram, is this any way to have different format like Circos or something that you can pack it in or visualized in form of paper? If you put it like half page, is it possible, for example? Thank you very much.
Okay, that's a lot of questions. Back to the first one. So you don't have to use Excel to format your data. That's just what Malcolm was doing in that example. Flapjack can handle very, very large data set sizes. I mean, obviously, ultimately, it depends on how much memory you've got on your machine, but we've comfortably had data sets with millions of markers and tens of thousands of lines. Obviously, you need to balance that against whether it actually makes sense to try and visualize data sets of that size. But obviously, with some of the analysis options and the ability to filter, then there might still be reasons for doing that. But I mean, just buy what you've got and see. I mean, you should be okay up to pretty big data set sizes.
I think currently we've got in Barley, our Barley database, we've got a data set with just over 11,000 lines and 50,000 markers. So we've got over 500 million data points, and Flapjack can handle that very easily. So I'm sure other people have bigger data sets, but that's what we're currently using.
So, for example, stuff we've done with Simit in the past, as Ian was saying, it's something like 1.4 million markers by maybe 30,000 accessions. So it takes a minute to load, but once it's in, it's fine. And then the grass pea and finger millet data sets we've got for this project are much smaller, so there's no issues there.
And obviously, if you start getting into generating similarity matrices over data set sizes like that, it's going to take a while. But I mean, Flapjack is multi-core enabled, so if you've got 16 cores on a laptop or a server, it will run much faster anyway for doing that kind of analysis. I'm trying to remember what your other questions were now. You asked about heterozygotes. It will draw if like literally if you have A/T, it's a little diagonal. Can you see if I've got a Flapjack here in the background that's got some share? Can you see that?
Yep.
Yeah.
Yep.
Find some heterozygote data here. Let's look. We just do highlight heterozygotes. We'll find some. There we go. So they get drawn like that.
Yeah. I saw. Yeah. Good.
In terms of dendrogram options, it's just using some R code behind the scenes on the server. So the image that we get back, Flapjack is just rendering that. Flapjack itself can't really do anything with it, but we can export the, obviously you've got the similarity matrix itself, and well, we can export I think we can do the tree from it, so you could theoretically put that into some other package to redraw it. And I can't remember what your fourth question was now.
Inferring missing data.
No, we can't do that. That's something we've thought about, some sort of imputation option. There was a package, I've forgotten its name now. We were looking at whether it's possible to sort of tie into that through an API to infer missing data, but it's something we need to look at how we would do that. But it's definitely something that's been on the to-do list for a while.
So now, if we have genotype data and phenotype data possible to use, even QTL, so association I didn't see, like in the demonstration today, possible of association markers with traits.
So we don't do association analysis. You can order, correct me if I'm wrong, Ian and Malcolm, you can order lines based on a phenotype though. So you can load in phenotypic data and you can visualize that in ways, but we're not doing any of the association analysis with that.
Okay.
Yeah, I mean, Flapjack was originally designed just to do visualization. It's only really latterly we've started to add some of these analysis options.
Yeah.
Any other questions that people have? I'm sure there'll be lots of questions once people start having a play about with it, of course.
So if it's still possible to ask question, I would ask, is this free we can download, or it's paid?
It's free, completely free.
I'll share a slide in a minute with a link to it, and you can go and get it. Yeah, so all our software that we develop here and at JHI is free for people to take and use.
Thanks.
Well, if there are no more questions, I'm going to put up another couple of slides.
There is, I see Bernadette.
Good. Bernadette, please.
Thank you. Yes, I have written a question in the chat about the heterozygous individuals. So I understand that because we are working on heterozygous species, so if most of my samples are heterozygous, it's not a problem, if I understand. So are the analyses available for this type of situations too? Are they relevant for heterozygous samples? And the next questions were about polyploid heterozygous, or the situation where samples are pools of individuals, because we are using this type of genotyping because we have synthetic accessions, synthetic populations. Thanks.
Yeah, so I mean, you're safer certainly for the analysis side of things because it hasn't been tested much beyond the diploid side of things. So long as it's diploid, even heterozygous data, you'll be fine. Beyond that, I wouldn't trust the analysis. Even though, I mean, you can load non-diploid data into Flapjack. It won't always render correctly, but yeah, it was designed around diploid data.
Does that help, Bernadette?
Yes, it's always helpful to know if a software is applicable to our situation or not. So, okay, so I understand we can test it for heterozygous diploid.
Heterozygous diploid will be completely fine, yes.
Okay, and then, but not for the other situations.
But not for polyploid.
Okay. So just a couple of last slides. If any other questions come up, just shout out. But just in terms of what's next, so we'll send you a copy. I've said these slides. There's not many, but there's a couple of links in here that will be useful. That we'll get to a bit, and we'll also send everybody else everybody who's been here a copy of the recording of this meeting, and you can share that with other colleagues and things who you think might be interested. The point in these training sessions was kind of like an introduction to get people's feet wet and dabbling in the area. So we're also keen to get feedback and things on what else we could do, maybe more advanced, going into things in greater depth and that kind of thing. And as part of that, Malcolm has spent a fair bit of time creating some videos. So they're quite short, maybe a couple of minutes, up to 10 minutes, and they cover some of the stuff we went through today, and they cover some other things in greater detail. And I would encourage everybody to have a browse through that. So the link's here. It will send this out, so don't worry, you don't need to write it down and get that to you. The other thing is, as part of these 3 trainings we've run so far, is we're just trying to get some feedback. So this is the first time we've done this. How do we make things better? These are quite short sessions, so maybe quite a lot is packed in. Maybe you want to see additional ones, whatever it is. And again, links here, and encourage people to go and fill that in if they can, and that allows us to make improvements. You can contact, I say us, but it's really Ian, by email. So there's a flapjack@hutton.ac.uk. Please, any questions, any problems, whatever, get in contact with Ian, and he's very quick to respond to things, and you'll get answers quickly from that. And lastly, there's a link here at the bottom which you can go to, which will provide all the help documentation we've got. There's some other resources and things I think we've got there, Ian, and as well as downloading the software so you can install it on your computers. And lastly, there was a paper in 2010 down at the bottom right here, which will give you some more information on Flapjack. I guess a lot of it will be out of date by now, but the general principles will still stand, so it's worth having a look at. Just to also highlight the fact that this was funded by the Templeton World Charity Foundation. We're working with GrassP and Fingermillet in that project, but it's also had input in the Templeton projects, had input from lots of other different places and funders as well. Of course, ICRISAT and ICARDA and the Crop Trust being the main ones that we've interacted with. So finally, thank you. Just again to reiterate the email address if there's any problems, and the link. Is there anything else anybody wants to ask or bring up just now? Nope, we're all fine. Well, in that case then, I'd just like to thank everybody for taking the time out of their days. I realise it's normal time for a lot of us, but I know Alan is probably going off to his bed straight after this. So just thanks everybody again, and we'll be in touch with all of the materials that went with this. And as I say, any questions, just give us a shout. So thanks everybody. See you soon.